Hey everyone, welcome. Today, we're going to embark on this incredible journey. Think of it like a detective story that took, get this, 150 years to solve. And what was the mystery? Well, it was figuring out our own biological instruction manual. We are talking about the history of genetics, the science that finally cracked the code of life itself.
All right, let's just jump right in with a simple but really profound question. What makes you, well, you? You know, the color of your eyes, how tall you are, the way you laugh, all the little things that define you as a completely unique individual.
For centuries, this was a total mystery. I mean, nobody had a clue. But now we know the answer is hidden inside a biological code. The story of finding that code and then figuring out how to even read it, well, it's this epic tale of forgotten geniuses, brilliant ideas, and world-changing discoveries. So, let's get into how we cracked that code.
So, our story doesn't start in some high-tech lab with fancy equipment. Nope. It starts in the quiet garden of a 19th century monastery. Seriously, it's the totally unlikely beginning for one of the most monumental sciences in all of human history. Meet Gregor Mendel. He was an Austrian monk, and through thousands, and I mean thousands, of meticulous experiments with pea plants, he figured out the fundamental rules of how traits get passed down from one generation to the next. He is, without a doubt, the father of genetics.
Okay, now what's really wild here is the timeline. Mendel publishes his revolutionary work in 1866, but the world just wasn't ready for it. It was completely ignored. He died in 1884, having no idea, not a clue, about the revolution he had just kicked off. It wasn't until 1900, a full 34 years after his paper came out, that his work was rediscovered, and the field of genetics was truly born.
So, the absolute core of Mendel's work was this. He proposed that heredity wasn't about blending traits, like mixing paint. Instead, he said it was controlled by these discrete, invisible factors that were passed down from parents to their kids. Today, of course, we call those factors genes. This was a totally radical idea at the time, and it laid the groundwork for everything that came next.
Okay, so Mendel knew something was being passed down, but he had no idea what it was actually made of. The next chapter in our story is all about the race to find the physical molecule responsible for heredity, a hunt for what scientists started calling the transforming principle. So for decades, scientists were chasing this mystery. They knew that some chemical substance inside cells could transfer traits from one bacterium to another, basically transforming them. So they gave this elusive substance a suitably mysterious name, the transforming principle. But what was it? Was it a protein? Was it something else entirely? Nobody knew.
And then the breakthrough. A series of brilliant experiments, culminating in 1952, finally proved it beyond a shadow of a doubt. The molecule of life, the carrier of our genetic inheritance, was DNA, deoxyribonucleic acid.
Now, just identifying DNA was one thing, but understanding how it worked was a whole other ballgame. And the famous discovery of its structure, well, it wasn't a solo act. It absolutely depended on the crucial work of others, like Erwin Chargaff, who figured out that DNA's chemical bases always pair up in a specific way, and especially Rosalind Franklin, whose stunning X-ray images provided the essential clues to its shape. And this is the result. In 1953, James Watson and Francis Crick put all the pieces together and revealed the elegant, iconic double helix. And it wasn't just a pretty shape. Its structure immediately suggested how it could store vast amounts of information, and crucially, how it could copy itself. The instruction book of life was finally open for business.
All right, so we found the book and we figured out its shape. The next huge challenge was to develop the tools to actually read the words on the page, and maybe, just maybe, even edit them.
The first major tool came along in the early 1970s with the discovery of restriction enzymes. The best way to think of these is like molecular scissors. For the very first time, scientists could cut a long strand of DNA, not just randomly, but at specific, predictable locations. This gave them an incredible new level of control. Then, in 1983, another game changer arrived, PCR. If restriction enzymes were the scissors, then PCR was the molecular photocopier. It allowed scientists to take a tiny, almost invisible amount of DNA and amplify it into millions of identical copies, plenty to see, to study, and to analyze.
You really can't overstate the impact of PCR. As the New York Times put it, this technique was so revolutionary that it basically split the entire history of biology in two, before PCR and after PCR. It unlocked everything from forensic science to disease diagnostics.
So, with these powerful new tools in hand, humanity was ready to take on its most audacious biological quest yet, to read our entire genetic instruction book from cover to cover. This was the Human Genome Project. And the sheer scale of this project was just mind-boggling. The goal was to sequence all 3.2 billion chemical base pairs that make up human DNA. It was this massive international effort that took 13 years and cost billions of dollars. Just think about that number, 3.2 billion. The results, which came out in 2003, were just full of surprises. For one thing, we learned that 99.9% of our DNA is identical to every other human being on the planet. We also found out that only a tiny fraction, like 2%, actually codes for proteins. And maybe most humbling of all, we realized we have no clue what almost half of our genes even do. Far from being the end of the story, the Human Genome Project was really just the beginning. It gave us the complete book, but the real work of understanding it had only just begun. So, why does this 150-year journey of discovery matter? Well, because cracking this code has fundamentally changed our world, and it continues to do so every single day. In medicine, it's led to targeted drugs, revolutionary vaccines like the mRNA ones we've all heard about, and tests that can spot our risk for certain diseases before they even show up. In agriculture, it's allowing us to develop crops that are more nutritious, that are resistant to drought and disease, which is obviously crucial for helping to feed a growing planet. And it's even become a vital tool in conservation, helping us protect endangered species, understand ecosystems, and fight back against the illegal trafficking of wildlife. So after 150 years, we've cracked the code. We hold our own biological source code in our hands. The journey to fully understand it is really just beginning, but it leaves us with the deepest question of all. Now that we can read the book of life, what new chapters are we going to write?